Hello, everyone, and welcome to today's webinar, Enhanced ACE2 Humanized Rodent Models to Make COVID-19 Research More Translatable. I am Sydney McNeil of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Envigo. To learn more, visit envigo.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I would like to now introduce our speaker, Dr. Zhao, head of the GEMS R&D team at Envigo. Dr. Zhao, you may now begin your presentation.
Good morning, good afternoon. Thanks for attending the webinar. My name is Justin Zhao. I'm from Envigo Genetically Engineered Models Services Group. Today, my topic is enhanced ACE2 humanized rodent model to make COVID-19 research more translatable. So as of today, there have been more than 260 million confirmed cases of COVID-19, including more than 5.2 million cases of deaths. The virus causing COVID-19 is called the severe acute respiratory syndrome coronavirus 2 or SARS coronavirus 2. The SARS coronavirus 2 is a single-stranded positive sense RNA virus. It has 78% homology to the original SARS coronavirus. Both the original SARS coronavirus and the novel coronavirus utilize their spike protein to interact with their cellular receptor ACE2 protein to enter the host cells. Another protein called the GMPRSS2 first activates the spike protein by proteolytic process. Secondly, after the spike protein binds to the ACE2 receptor, the GMPRSS2 can further cleave ACE2 protein to facilitate the facilitate the fusion of viral and cellular membrane to promote the virus uptake. According to the structure data, the human ACE2, those human ACE2 residue directly interact with the spike protein. Compare those residue with rodent ACE2 residue. We can clearly see the homology between human ACE2 residue and rat and mouse ACE2 residue at the binding site are not very high. So therefore, the wild-type mice and rats cannot be efficiently infected with SARS-CoV-2 virus. To overcome the low affinity issue, there are a couple of different strategies that will be used by different groups. The first strategy is use most adapted SARS coronavirus. Basically, pass the virus into the wild-type mice to get the more than 10 passage to get the most susceptible virus variants. Mainly mutations, find mutations in the receptor binding domain of the spike protein. While those strategies can directly utilize most endogenous ACE2 protein, it cannot directly test the different variants of virus because each time we need to pass the virus again in the mouse again. So the second method is use replication deficient virus to transiently express human ACE2. For example, use adeno virus or use adeno-associated virus to mediate human ACE2 expressing, typically packing a ACE2 expressing cassette into a virus. Most likely use a ubiquitous promoter like a CMV promoter. Then infect the animal with adenovirus or adeno-associated virus to express to translate express human ACE2 protein in the lung. This strategy, is not convenient. Each time you need to infect first with adenovirus or adeno-associated virus, then infect with actual SARS-CoV-2. And for each animal, you need to infect it individually. So most widely used method is use human ACE2 transgenic mice. There are different types of human ACE2 transgenic mice. The most widely used one is called the K18 human ACE2 transgenic mice. Those mice basically use human ACE2 or human carotene 18 promoter to drive with a type of alpha mosaic virus in cancer to drive human ACE2 expressing in epithelial cell, including airway epithelial cell. In the original model, the transgene copy is 8 copies, and the original test showing the mortality rate can be as high as 100%. The second type of transgenic model is use another promoter called the HFH4 or FOXGen1 promoter to drive human ACE2 expression. Those promoter is called lung-ciliated epithelial cell specific promoter. But in the original model, the author also shows it's also expressed in different organ or tissues, including brain, liver, kidney, GI tract. Copy number is unknown, but mortality rate is roughly 50%. The other transgenic model is use a synthetic promoter called the CAX promoter or CIG promoter. We know a CIG promoter is a very strong synthetic promoter with a CMV in cancer. Its expression is ubiquitous expression, basically every tissue where expressing human ACE2. So overall, those models support SARS-CoV-2 infection and also induce lung inflammation and damage. But the major drawback is high mortality rate caused by neuroinvasion and encephalitis, probably due to very high levels of human ACE2. Additionally, those mice still express mouse ACE2 protein. So ideally, a model expressing physiological level from human ACE2 at mouse endogenous ACE2 expression cell will be better. This can only be achieved by using a human ACE2 Knockin model, not the random transgenic model. So to make human Knockin model, we utilize the CRISPR-based technology to mediate the integration of the human ACE2 cDNA into the mouse gene locus. So specifically at the coding ATG site. So this way can ensure human ACE2 expression is under control of mouse ACE2 promoter and the regulatory element, whereas mouse ACE2 expression is inhibited. So we micro-injected the CRISPR reagent along with the donor DNA into fertilized one cell stage embryo and screen the founder animal with 3 different internal PCR primer sets, as well as 2 different junction PCR primer sets to ensure those integration is complete and also at the right location. We found 2 founder animals. To check the human S2 expression, we first use some air tissue to check the human S2 mRNA expression by RT-qPCR. We designed 3 sets of primer or probe targeting to different regions of human S2 cDNA. Yeah, then cut some S2 the air tissue from both wild type and Knockin mice to save the animal. You can clearly see all those 3 probes can recognize human S2 mRNA. As expected, the control will not show any mRNA express human S2 mRNA expression. Yeah, later we systematically examine the human S2 expression by RT-qPCR to check the different organs. The first organ we check is lung. As you clearly see, the hemizygous animal and heterozygous expresses human S2, whereas a wild type of control, male or female, do not. By the way, the S2 gene is an X-linked gene. So for male animal, they only have 1 copy. It's called a hemizygous. For female, can be either heterozygous with just 1 copy or homozygous with both copies. We also check the brain, and here we also include homozygous animal, female, yeah, hemizygous, homozygous, heterozygous, and both show expression of human S2. Yeah, we also check the other organ, including kidney, lung, stomach, basically those hemi, homo, and het all express human S2 as expected. We also check the mouse S2 expression in those tissues. As expected for hemizygous or homozygous, those endogenous S2, mouse endogenous S2 expression will be disrupted. You can clearly see the expression level for hemizygous and homozygous, but the mouse S2 is greatly reduced compared to wild type, male, female, or male. They are in different organs, from lung, brain, kidney, heart, and stomach. Yeah, we further confirm the human S2 expression by a Western blot. So we first choose kidney tissue because kidney tissue is easy to harvest, and according to literature, kidney express high level of S2. We use a human S2 specific antibody. Check animal from both hemizygous, homozygous, and hemizygous from 2 different lines. Now we can clearly see there is a human S2 band at the expected molecular weight size. We also use lung tissue, and although the variation, we saw large variation among different animals, but we can clearly see in human S2 protein is expressed in the lung. Yeah, there was a report published in Cell showing that human S2 gene, but not the mouse S2 gene, is a interferon stimulate gene. The SARS coronavirus could exploit interferon driving upregulation of S2. Basically, once a virus infected the cell can express, host cell can express secrete the interferon, and the interferon can stimulate the S2 expression, so the virus could take advantage of those immunodefense system to boost each cellular receptor expression. So for this reason, we also create a model called using human gene containing a DNA construct which contain entire human S2 gene, including promoter sequence, as well as some neighbor gene. But the neighbor gene is just a partial of the neighbor gene without the promoter, so the neighbor gene will not express. So we microinjected the BAC DNA construct into put like one cell stage amber and screen the founder animal with different primer pair targeting the different region of the BAC DNA construct. As you can see from here, there are at least 3 founder were developed. So we also check the human ACE2 expression in those transgenic mice by Watson blot. Here are a sample from 2 kidney sample from 2 founder line 6 and line 11, and clearly see the human gene ACE2 is expressed. So in the beginning, we mentioned the TMPR has can prime the spike protein and also facilitate the virus uptake. So for this reason, we also created a TMPR ACE2 Knockin model. Basically, use the same strategy, CRISPR-based strategy to mediate the knocking of human ACE2 cDNA into the mouse TMPR ACE2. Sorry, not the TMPR. Yeah, TMPR ACE2 into a mouse TMPR ACE2 gene locus. Here we did not pack the phosphodieting exon because phosphodieting exon only codes for 5 amino acids, and there is no difference between human and mouse TMPR in the first 5 amino acid radio. So we packed the second coding exon, but this study still can show human ACE2 cDNA expression is under control of mouse TMPR ACE2 promoter and the regulatory elements, whereas the mouse TMPR ACE2 expression is disrupted. Yeah, use same strategy. We microinjected the CRISPR reagent along with the TMPR ACE2 donor DNA and screen the founder animal with internal PCR primer as well as junction PCR primer. And here the sequence product also was confounded. PCR product was also confounded by sequence. Here we can see one founder animal was identified. We also check the human TMPR ACE2 expression by RT-qPCR. Those just from heterozygous animal. So of course, the mouse ACE2 is also expressed. Yeah, we sent some human ACE2 transgenic as well as the human ACE2 Knockin model to our collaborator at the Georgia State University. They infected those mice with SARS-CoV-2 virus through intranasal route. Then they monitor their animal weight daily after virus infection. From here, you can clearly see both hemizygous, which is blue, and homozygous animals start to significant weight loss after day 4, so the first day after virus infection. And specifically, the hemizygous animal lost too much weight, so our collaborator has to euthanize the animal at day 9. Whereas the hemizygous animal, the homozygous animal somehow still survived the entire test time, which is 2 weeks. In contrast, the transgenic line did not show any significant weight loss. Instead, it shows slightly increase, probably because of the age increase. So our collaborator also checked the antibody level in those animal that survived the entire test. So there are 3 animals. You can clearly see the express some antibody against the human, against the SARS coronavirus. And the transgenic line also express some antibody, but they're just at low level. Our collaborator also checked the virus RNA by RT-qPCR from the for hemizygous mice sacrificed after day 9, as you can see from here, the virus did present in both brain and the lung at different concentration. So in the beginning, we mentioned that the one drawback of KT model is very high level of virus or brain damage. So the virus in the same group that also published a paper use the same method. Use the KT model. And from KT model, you can clearly see the virus in the brain load is much higher than what we discussed here. Here is 10 to 6 at day 9. And in here, even day 6, you can see the virus is 10 to 10. So at least we don't need to worry about the virus cause the damage, brain damage for the Knockin line, because the virus load in the brain is at least 1,000 times lower than the KT model. Those work was done by the same group. We also sent our mice to another collaborator, Dr. UAB, University of Alabama, Birmingham. The infected mice, those are just the Knockin mice with different age. So what I labeled here is 103 means 1 year 3 days, and it's a female, so just 103D and F. From here, you can see there are 2 mice. One is this one with the age of 177 days. It's a male, and another is 103 days age, and it's a female. They shows significant weight loss at the day 7, so they have to euthanize those animals. The other animal, some see significant weight loss, but gradually recovered and survived the entire 2 weeks test period. They also use some histology analysis to check the vascular epithelial and the vascular structure, and they found some alveolar epithelial and the vascular structure is changed or damaged by the virus infection. And I also found that immunocell infiltration is a CD45 mark. We also made a human ACE2 Knockin rat model, because rat is a preferred model for some applications because of its large size and also better neurological behavior or cognitive behavior. So basically, we use the same method. It's a CRISPR-based method, knocking human cDNA into the host-coding exon of rat ACE2 gene locus. Also screen the founder animal with both internal PCR primer set and junction PCR primer set to ensure that particular integration is completed and also at the right location. During here, it's clear we got the 6 founder animal, and still use same RT-qPCR method, check the human ACE2 expression with 3 different problems. Here is use different tissue to systematically examine the human ACE2 expression in hemizygous, homozygous, and heterozygous, all the 5 tissues. And clearly see the human gene is expressed in those organs. We also check the rat ACE2 expression, and as expected, in the lung tissue, those hemizygous and homozygous rat ACE2 is not expressed in those homozygous or hemizygous animals, which is correct. Also in the brain, Also, we did not see rat ACE2 expression in hemizygous, homozygous, and same is true in stomach. But somehow in kidney and in heart, there's still some level of expression. Probably because we used rat ACE2 gene is not very specific, could recognize some other transcript or mRNA from kidney and heart. We also check the human ACE2 expression by Western blot. You can clearly see the human ACE2 is expressed, or the band is presented at the expected site. The sample is a kidney sample. Yes, we also made a human ACE2 BAC-based transgenic rat model. Basically, it's use same BAC DNA constructor. This constructor including entire human ACE2 BAC gene, including promoter and some neighboring sequence. And we screen the founder animal with a different set of PCR primer to target the different region of that construct. That we also check the BAC expression. You can clearly see a human ACE2 gene is expressed in those three tissues in the lung, kidney, and the brain. We also include the wild type animal as a control. And those is a rat endogenous ACE2, because for BAC transgenic those will not affect endogenous ACE2 expression significantly. So they are still expressed here, rat ACE2. So we also confirm the human ACE2 expression in those transgenic rat with Western blot. You can clearly see the human ACE2 is expressed in the BAC transgenic line. There's still a kidney sample. So basically we have developed the humanized ACE2 mouse and rat Knockin model, as well as human ACE2 gene-based BAC transgenic mouse and rat model. Expression for human ACE2 in those mouse and rat model was confirmed by RT-qPCR and Western blot. And the humanized ACE2 Knockin mice challenged with SARS-CoV-2 showed weight loss and other phenotypes. And the humanized TMPRSS Knockin mouse has also been established, and expression of human TMPRSS2 was confirmed by RT-qPCR. So, I'd like to also thank my colleagues who contributed to this, particularly the Genetically Engineered Model and Service group, and also our marketing group. And I also like to take this opportunity to thank our collaborators at the Georgia State University, and also our collaborators at the University of Alabama at Birmingham. Thank you for your attention. If you have any questions, if I don't have time to answer you, please email me. Thank you.
Thank you, Dr. Zhao, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, could you please explain why your human ACE2 gene-based BAC transgenic line did not show obvious weight loss in virus challenge test?
Hello, thank you for this good question. We actually don't have a good answer for this. We are still trying to figure out this as well. According to the serial paper, the human ACE2 promoter is interfering stimulated promoter. So we hope the human ACE2 transgenic promoter-based transgenic model will also have some phenotype, but somehow the animal we tested did not show weight loss. But I have to explain the animal we tested all from one founder. So we also have other founder from animal from other line we are going to test again. But if we still see similar result, the hypothesis will be probably the mouse interfering cannot stimulate all the human ACE2 promoter somewhere efficiently. So that's only explanation. Thanks for, this is a good question.
Perfect, thank you. Our next question is, post-infection with the humanized ACE2 mouse model, have you checked viral load presence in the lung at early time point? And if so, at what days post-infection and what was the load?
This is a very good question because virus load, according to literature, in lung is gradually reduced. The data we're showing here is at the day 7 post virus infection. The load is not the highest point. Our collaborator did check the older point, and they show about 1,000 times higher what we listed here. We just got the most recent update last week, so did not include those data in the presentation. Sorry for that. But did see high virus load in start from day 3 to day 5. Thanks.
Wonderful, thank you. Our next question is, can you compare and contrast your ACE2 Knockin model with the K18 ACE2 transgenic mouse model?
This is a big question. It's a good question. I think that both model can support the virus infection. The difference is K18 model currently is widely used because of those model was generated on original SARS coronavirus disease. Its expression level is relatively high because it's a random transgenic with a copy of the human cDNA. The promoter for ACE2 or K18 model is human Keratin 18 promoter. It's different from mouse ACE2 promoter. So the difference is for transgenic model with human Keratin 18, there was expressed some human ACE2 protein. In some cells that normally do not express mouse ACE2. So that is one thing. Secondly, because of the high copy number, probably explains why the mortality rate is so high. Additionally, those animals still express mouse endogenous ACE2 protein. So those will affect the final data interpretation. So in contrast, the Knockin model, as you can see from the presentation, the endogenous protein is completely disrupted or is blocked. So that is much better. And the expression level in Knockin model is physiological level. So the any data will be got from Knockin model will be more relevant to human disease. But for KT model, still useful. For example, if you just test some vaccine, you can use a boost model. Either model will be okay. Thanks. That is a good question.
Perfect. Thank you. Our next question is, have you tested the effect of human TMPRSS2 on the SARS-CoV-2 virus infection?
This is a good question. From the beginning slide, we said that both S2 and TMPRSS2 play important role in COVID-19 disease. We made both Knockin model, but currently we haven't tested those so-called double Knockin model. We're still in breeding stage and will be available somewhere a couple of months. So we haven't tested, but it will be very interesting model.
Thank you. Our next question is, in your ACE2 Knockin model test, have you checked injury in other organs after SARS-CoV-2 infection?
This is a good question. Yeah, our collaborators are doing those in other organs, including some test some cytokine profiling. Yeah, we haven't got their updated. We will have their data soon. Yeah, they're looking at the other organs and other marks as well. Thanks.
Perfect. Thank you. Our next question is, are all of those models you discussed currently available?
Most of them are available. As I mentioned, that some will be we don't have too many animals available. Some will have many animals available. So for double Knockin, for TNPR ACE2, our focus is generate both TNPR Knockin and human ACE2 Knockin in one animal. So still in breeding stage. Yeah, thanks.
Thank you. Our next question is, what SARS-CoV-2 variant did you use for your challenge studies?
That's a good question. Yeah, the one used here is just the original strain. Yeah, we haven't tested. We are going to test a different virus strain as well. Including the Omicron.
Perfect. Thank you. Our next question. Have you performed any pathological analysis in your models after infection?
Yeah, we did not do much of this because all our collaborators are doing this as work. Yeah, we just got the data. The only histological data we is showing in the slides. But we have two collaborators that are working on that. Thanks.
Thank you. It looks like we have one final question. Can the high reinfection rate in new SARS-CoV-2 variant be explained by higher ACE2 expression in patients?
I don't think so. In higher ACE2 expression, probably in human patients, the ACE2 expression level will be same. The new variant is probably somehow binding more efficiently to the human ACE2. But anyway. Yeah, it's a good question, and we should check the new variant as well. Yeah. Thanks.
Perfect. Thank you. Thank you again, Dr. Zhao, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Envigo, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.